In the previous video, we saw the different stages of photosynthesis. And now in this video, we will be discussing the location of photosynthesis, which place in the plant the photosynthesis is taking place. We all know that photosynthesis is dependent on a particular pigment, and the pigment is called the chlorophyll pigment. Now we all know that the chlorophyll pigment are abundantly found in the cells of the leaves. So leaves have the particular specialized parenchymatous cells, which are called as the mesophyll cells, and they carry enormous number of this chlorophyll pigment. And in class 9 you have studied about different cell organelles. In that you have studied the chloroplast is the cell organelle which is found only in the plants which contain this particular protein pigment, which is called as the chlorophyll pigment. Now, for understanding the chloroplast better, first let us understand the leaf TS, the transverse section of a leaf, and which are the cells which actually contain this chlorophyll pigment. Now I have drawn the transverse section of the leaf, a place where the photosynthesis is taking place. So I'll be labeling it, and each part we will be discussing. So this part is the cuticle. The cuticle is a waxy covering for the leaf. It provides protection from any kind of external damage. Now next to the cuticle, these cells are called as epidermis, the outermost layer in the leaf tissues. The outermost tissue layer is called epidermis. Now next to the epidermis, the next layer, the little bit elongated pyramidal shaped cells, which are called as the mesophyll cells. So these are special mesophyll cells, or these are of parenchymatous in nature. So they are called as the palisade mesophyll. These are called as the palisade mesophyll. And the cells which are present here, these are called as the spongy mesophyll cells. Why are they called spongy mesophyll cells? Because they contain more amount of air spaces in between the cells. So they look like a sponge. They are spongy in their appearance or texture. That is why these mesophyll cells are called as the spongy mesophyll cells. Whereas the more connected cells found in the upper layer, they are called as the palisade mesophyll cells. So both are parenchymatous in nature, right? So I will write it as the spongy mesophyll cell. Okay? The spongy mesophyll cell. Now this is the vascular bundle. The vascular bundle. Now what is the vascular bundle? The vascular bundle contains a collection of vascular tissues. Now which are the vascular tissues? The conductive tissues, the transporting tissues are called as the vascular tissues. So which are the conducting tissues? You have studied in class 9. The conductive tissues are xylem and phloem. So xylem and phloem location in the leaf will be here. And these are the mesophyll cells, and this is your lower epidermis, and in the lower epidermis there are presence of tiny pores guarded by kidney bean shaped cells, and that structure is called as stomata. Stomata is present in both the epidermis, upper and lower, but more numbers are found in the lower epidermis. Now, in this structure, the palisade mesophyll cells are the majority of the time, the palisade mesophyll cells, they carry out the photosynthesis. and in some species of the cells, the spongy mesophyll will also carry out photosynthesis. So, major amount of the chlorophyll pigment that is present inside the chloroplast cell organelle are found in this palisade mesophyll as well as the spongy mesophyll cells, right? Okay, now once we have now discussed about the structure of the leaf, now let us discuss the structure of the particular cell organelle which contains the particular pigment, the most important pigment, that is chlorophyll, which has the capability to absorb the sunlight. Why chlorophyll is important? It absorbs the sunlight. It has the capability to trap the sunlight. And after trapping of sunlight, only the rest of the events of photosynthesis can take place. Like the absorption of sunlight is the first stage, then chemical energy, light energy is converted to chemical energy, then splitting of water, then comes the reduction of carbon dioxide. So let us just see how the cell organelle, you have already studied in class 9 about the chloroplast. Now let us again draw that and see how the structure of chloroplast is there and how it facilitates the function of photosynthesis. So now let us draw the cell organelle which is called as the chloroplast. So chloroplast is a double membrane structure. Okay? Double membrane structure means it has two membranes. It has two membranes, one the outer and the inner membrane. Okay? So there are many organelles which have double membrane. So chloroplast is one such organelle. Apart from that, we have Golgi bodies, we have mitochondria who are having two membranes, one outer membrane and one inner membrane, and inside the inner membrane there will be a matrix like structure, a jelly like fluid like structure, right? So here also the fluid like structure is present, and that fluid like structure is called as the stroma. So the jelly like or fluid like structure which is present in between, inside these two membranes, is called as stroma. Now inside the stroma there are stacks of disc shaped structures. Okay, there are stacks of this disc shaped structures, and we will be labeling it, this disc shaped structures, right? Okay, so these stacks are present inside the stroma. Now what are these stacks called? These are called as the grana, right? Okay, if only one such stack we take into account, then we will call it as granum. Granum is the singular. Singular is granum. That means when I am talking about only one of this, I will refer it as granum. But I have drawn two lines. That's why I have written here grana. Now the grana, the stacks which is the disc shaped structure which actually make the stack, they are called as thylakoid. So this disc shaped structure, I will label it as thylakoid. Okay, which one is the thylakoid? This coin shaped structure, the disc shaped structures are called as the thylakoid. So now when we talk about each granum, a single granum is made up of different, like many stacks of, many numbers of thylakoid stacked together, right? When they are stacked together, that combinedly structure is called as granum. Granum is made up of stack of thylakoid. Which are the thylakoids? These disc shaped structures are called as thylakoid, right? And I have drawn some lines here, these lines, right? So what are these lines? These lines are called as stroma lamellae. Okay, the lines in between the stacks, joining the stacks, is called as stroma lamellae. Now, the thylakoid is the region of light reaction. Okay, so thylakoid carries out light reaction. Okay, so light reaction is carried out by thylakoid. So where is the location of light reaction? Now this disc shaped structures are the location of the light reaction. Now what are the events that is taking place in light reaction? They have to trap the solar energy, the sunlight, and then first stage is over, trapping, absorption of solar energy. Now next what happens? Conversion of the solar energy, the light energy, into chemical energy, and at the same time what happens? Splitting of water molecules, right? So these two events that take place in the second stage, right? First stage is the absorption of light energy, trapping of light energy. Stage number 2 is the conversion of light energy into chemical energy, and at the same time the splitting of water molecule also takes place. Now after this process, the next phase is the dark reaction. What happens in the dark reaction? The carbon dioxide is reduced to carbohydrate, right? The reduction of carbon dioxide into carbohydrate. So this is the third stage, and this comes under the dark reaction. So which is the location of dark reaction? The stroma is the location of dark reaction. Okay, so this gives us the detailed idea of the structure of chloroplast. So once the light reaction is completed, then only the dark reaction can be proceeded. But in some plants what happens, it does not mean that always the dark reaction will follow the light reaction. Some of the xerophytic plants, like the desert plants, they follow a little different method of photosynthesis, as they have the fear of losing more water during the daytime. So what they do, they keep the stomata closed during the daytime. So during night time only they open the stomata and they take the carbon dioxide inside the cell. Now the carbon dioxide inside the cell follows some biochemical reactions and gets converted into another product, and the product is stored in the cell, right? And the next day when the sun rays comes, the sun rays will be accepted and the light reaction as well as the dark reaction will be completed. But during the daytime there is no exchange of gases, there is no absorption of carbon dioxide, or there is no intake of carbon dioxide, because they do not open the stomata. The stomata is not only there for gaseous exchange, it is also there for transpiration, the process of losing excess of water. But when it is the question of desert plants, they cannot afford a lot of loss of water from their plant body. So they follow a different method to cope up with the surrounding situations. Now let us understand the structure of stomata and the role of stomata in the photosynthetic process. Stomata is a tiny pore. Already we have discussed the tiny pore which is present mostly in the lower epidermis of the leaf cells, the epidermal cells of the leaf, and these pores are surrounded by a kidney bean shaped cell. They are known as the guard cells. Why are they called the guard cell? Because they guard the pore, the opening and closing of the pore is guarded by this bean shaped cells. That's why they are called as the guard cells. So let us draw the structure of stomata here. Okay, so now this is the stomata, stomatal pore, and these are the guard cells. The nucleus of the guard cell and the presence of chloroplast. So these two are guard cells. Okay, so combinedly these two structures are called as the stomata. So now what is a stomata? Stomata is a collection of two things. One is the stomatal pore, the pore in between the guard cell, or the pore made by the two guard cells present adjacent to each other. Now apart from that stomatal pore, we have guard cells. So combinedly these two structures, they form the stomata. Now from where these guard cells are coming? They are not coming from somewhere outside. These are the modified epidermal cells. So some of the epidermal cells will get modified into the structures, the kidney bean structures, and they are termed as the guard cells. Now what is the role of these guard cells? The guard cells almost control the opening and closing of the stomatal pore, right? So how do they control the opening and closing, right? So why we, first of all, before going into that, let us first understand why there is a need of opening and closing of stomata. So stomata cannot be open all the time because it has got a purpose. Stomata, the purpose is, first function of the stomata is gaseous exchange, and the second most important function is transpiration. Okay, and this particular structure which maintains the temperature, because once transpiration is its function, then it will maintain the water level, it will maintain the temperature, it will maintain the water loss. Everything is related to stomata. Now these are the two most important functions of this particular structure. So all the time the gaseous exchange cannot take place, all the time transpiration also cannot take place. So sometimes the stomata has to be in closed condition, sometimes the stomata has to be in open condition. So that is the reason there is a question of opening and closing of stomata. So when there is a requirement of gaseous exchange, the stomata has to be in an open condition. When there is a requirement of transpiration, the stomata has to be in an open condition. So when the plant wants to stop the transpiration, when the plant does not require the gaseous exchange, why it is going to keep open the stomata? It has to close the stomata. So the closing and opening gate is guarded by the guard cells, right? So the guard cells, they control the opening and closing of stomata. So when it is needed, it is open. When it is not needed, it is going to get closed by the guard cells. Okay? Now, how exactly the guard cells control this closing and the opening of stomata? Now, these guard cells, they are connected with the mesophyll cells. Okay? These are the mesophyll cells which connect the guard cell. So whenever the guard cell bulges by the entry of water from the surrounding cells, suppose the surrounding cells, they provide the water more into the guard cell starts bulging. You have read about osmosis, the principles of osmosis. Water will move from its higher concentration to the low concentration area. Suppose the guard cell has lost water by transpiration, then what will happen to the guard cell? The guard cell has less concentration of water molecule. Now what is going to happen? The water from higher concentration area will try to move into the guard cell. Now when the guard cell is filled with more and more water, coming from the higher concentration area, the guard cell, what is going to happen when a cell is there and water enters into the cell? What is going to happen? You have studied in class 9. What happens to the cell? What happens to the dry grape which you studied in the class 9 book? The dry grape, when it is put in the water, the dry grapes starts swelling, right? Why it starts swelling? Because there is entry of water molecule into the dry grapes. So same thing happens here. The guard cell receives water, so slowly it starts swelling up. When it starts swelling up, the two guard cells that move apart from each other and they open the stomatal pore. So when the guard cells are in a bulged condition, they open the stomatal pore. If the guard cell starts losing the water, it has lost the water by transpiration, then now what is going to happen? It will again close, because after the losing of water happens, the guard cell will start shrinking. This process you have studied in class 9, when the cell will shrink and when the cell will swell up. Water enters, the cell will swell up. Water goes out of the cell will shrink. So when the cell is shrinked, then the guard cell loses its turgidity, the thickness it loses, the tightness it loses, and it starts shrinking, and both the guard cells will come together. Clear? So they come together and they close the stomatal opening. So I'll just draw that also. When the guard cells, they are not swollen up, what is the structure? How the stomatal structure looks. So these are the mesophyll cells which are surrounding that, and the epidermal cells. Okay, so epidermal cells will be here, and this is a closed stomata. Closed stomata. Okay, so this is a open stomata. Clear? So this is closed stomata. Right? So guard cells, after losing the water, they shrink, they come together, they close this opening. So the stomata plays the important role in gaseous exchange. That means the carbon dioxide is taken in for photosynthesis and the oxygen is given out during the photosynthesis process. Apart from this, the stomata also plays the role in respiration process, where the opposite happens. That is, the oxygen is taken in and the carbon dioxide is given out. So all these functions, the most important role played by the stomata. So it helps for the raw material to enter into the plant. So now we have understood how the raw materials are coming into the plant, or what are the raw materials already present in the plant, and how collectively they carry out the function of photosynthesis. This produces the carbohydrate elements for the plant. Now, apart from carbohydrate, the plant also requires the other elements like the macro and macronutrients, which eventually forms the protein elements, the protein parts of the plant. So any protein molecule, any protein compound is to be synthesized. For that, the plant needs nitrogen. So you have studied in class 9 the nitrogen fixation. So this is carried out with the help of the bacteria rhizobium, the nitrogen fixing bacteria in the root nodules of the leguminous plants, and the nitrogen is taken in by the plants in the form of nitrates and nitrites. These are the absorbable form of the nitrogen. So the atmospheric nitrogen, it cannot be taken inside the plant. It cannot be absorbed directly from the atmosphere. That is the reason there is a requirement of fixation of nitrogen. So the plant takes the help of the microbe, the bacteria, and then the bacteria converts it into the nitrates and nitrites. Apart from that, the lightening process also provides the absorbable form of nitrogen that is happening in the atmosphere. And apart from that, one more method is also there, that is the industrial method, by which the nitrogen is fixed in the form of ammonia and it is given in the form of fertilizers. So this is how the plant gets all the elements and it synthesizes the required elements for its body.